Okay.
Yes. Okay. Test. Sound. And you have to close your. Oh yeah. No sound. No mic. No sound. Test. Pour couper les micros et les sons aussi.
Ouais.
C'est bon. C'est vert. C'est les micros.
Ah oui, c'est la salle.
Alors, test.
Le guest est comme Il y a des flags, n'est-ce pas?
Ouais, je vais avoir de.
Mais, ok. Avec Zoom, avec YouTube, ça va. Par contre, pour Zoom, est-ce que c'est assez rapide? Il n'y a pas de lag, non?
Il va avoir, certainement, il va avoir un petit lag.
Ok, ça va. Sur Zoom, ça va, en fait.
Ouais.
Ok, sur YouTube,
Yeah.
ouais, c'est ça. C'est
Ouais,
normal. Ok, ça va.
YouTube works.
Well, yeah. There is a bit of a lag.
Do you have sound on your presentation?
No, I don't.
No.
Yeah. Yeah. Good. Okay, perfect.
Okay. On est là, on peut utiliser du chien. Puis on en parle juste à voir si YouTube ça marche bien. Les gens écoutent sur YouTube aussi.
Alors,
Je peux l'ouvrir pour voir. Je vais tester si ça va bien fonctionner. Hey!
bien, je tente de voir.
Sur YouTube ici, je vais
Alors,
fermer la.
là, il y a marqué Carrefour zéro face, là.
Voilà, comme ça.
Tu viens de fermer.
Et les sons sortent là-bas. Vous allez faire un live streaming sur YouTube.
Ouais,
Là.
avec la chaîne de Brain. Et en fait, on avait déjà prévu la diffusion, donc il y a deux liens. Et donc, le premier lien, j'ai mis en titre
Allez sur
New link in description.
Et
du coup,
ça va être un jeu de piste en fait.
c'est ça. Parce que j'ai plus le temps de réannoncer
QR code.
Ouais. Pas le temps de- Ouais, mais sur la thumbnail, ouais, non, là, je peux pas faire en plus. Si quelqu'un d'autre veut le faire, parce que je suis pas le seul à avoir accès. Euh, Ouais, je voulais voir, je voulais juste confirmer avec toi.
Ouais.
C'est à 9 heures.
Ouais, c'est ça.
Ah, ouais, ok.
Décalé dans le temps.
Oui, non, mais là, on est à 16 heures.
Mhm.
Et bah, moi, tu vois, enfin, je sais pas, dans les journées avec les après-midi, forcément, j'ai le stress. Pour vous avoir en même temps avec Karine, je suis sur un montage. Ouais, tout ça, mission. Exactement, c'est synergie.
Ah ouais. Mhm. Ok.
Pour mettre.
C'est ça, des beaux.
Ok, début en fait.
Ouais, c'est ça que
Ok.
tu veux faire.
Ouais, c'est ça. Éligible en plus au Canada maintenant.
Ouais, je sais. Pour ça, je pensais.
Franchement.
Ouais.
Bonjour.
Et Julia.
Bonjour, Julia.
Bonjour. Euh,
C'est bon? Ouais.
ben moi, en fait, j'ai aussi avancé sur.
Mis comme ça.
Je vais déposer un.
Ok, ok. Enfin, les limites, c'était ici. Ok, ok, parfait.
Gros projet de ici, comme les rois.
Ok, merci beaucoup.
Ouais, ok. Ah oui, oui.
En EG, donc justement, j'ai bossé, j'ai un 3 pages.
Ouais.
À la fin, ça va faire 12 pages. Donc j'ai bossé cet été
Je
là-dessus.
sais pas, Julia, encore.
On s'est jamais croisés, je crois pas.
C'est pas vrai.
Non,
Ah,
on
mais
ne
tu nous
s'est
entends
pas
bien.
Eh ouais, elle est là. Elle nous entend.
Salut,
Je vous,
enchanté.
je vous entends très bien. Ne dites rien de compromettant parce que
Non, je sais, je t'ai entendu arriver. Donc, c'est une éroteur de
Ouais, ouais.
ouais. Et du coup, c'est elle qui est pas mal sur la structure, enfin, qui structuré toutes les différentes branches à partir de ce
Ben,
projet.
ben là, justement,
On va
le
rencontrer
projet,
les gens de,
ça va être.
Big Brains bientôt là aussi. Tu
Big
connais
Brains.
Brains, ouais. C'est la suite de trucs européens.
Ah oui,
Donc Big Brain, je sais plus comment ça s'appelait.
c'est Catherine Amons qui a repris ça, non?
Au niveau européen peut-être, mais au niveau
Ouais.
France, on
C'est
discute
qui?
avec enfin, au niveau Jean-François Langin
Ah, ok.
et
Ouais,
Philippe Vernier.
je connais bien Jean-François.
Tu connais, ouais. Et les gens qui sont intéressés par Rêve, en l'occurrence, c'est Andrea Bovelli et
et
Olivier
Olivier
David.
David.
Ah, Olivier David, je le connais bien.
Olivier
Vraiment.
David, ouais.
Mm.
Olivier David. Apparemment,
Un gros nom.
ils veulent faire la même chose en SEG au niveau
Ouais,
européen.
nice.
Voilà, on est en discussion. Enfin, on va rentrer en discussion là-dessus avec eux.
Ok. Ah, nous, on fait du SE, j'ai aussi à
Ouais,
Saint-Justin. Maintenant.
ouais, ok. Là, il enfant.
Ouais.
Bah oui, tu veux dire.
Ouais, non, justement, je vais faire du développemental.
Bonjour.
Salut. Ouais, ouais. Après, les volumes sont très faibles. Hein.
T'as vu un peu sur le.
Ben, ça va marcher.
Ça va marcher, ouais.
Mais justement,
Les volumes sont faibles, ouais, parce que du coup, Olivier David nous a parlé de volumes.
ben, nous, ici, à Saint-Justin, c'est faible. C'est genre un gamin tous les 3 mois, quoi.
D'accord.
Bah,
Mais,
Olivier David, il nous a dit qu'il a probablement le plus grand dataset, mais il nous a pas donné de chiffres.
ok.
Mais il travaille depuis des années là-dessus.
Ok.
Tu le connais bien?
Je le connais pas super bien, mais il était au Lénard, en fait, avant.
Ouais. Non,
Il a
il a
beaucoup
marqué.
travaillé avec les gens, mon ancien labo à Paris, bah, du Karim aussi, d'ailleurs.
Ok.
Avec bah, toute la clique à l'ancienne de Varela, avec
Ah oui.
Michel Levan Cuyenne.
Ah oui, d'accord. Ok.
Euh,
Ok, ok.
mais ouais, dans cette bail que j'ai pas vu. Je crois que je l'ai vu la dernière fois, c'était en 2020. Ouais. Donc, c'est 6 ans.
Ok.
Ça va, Venkatesh, t'as bien dormi?
Ouais, on veut dire vrai. Mais c'était pas mal en fait. C'était pas mal. J'ai dormi pendant 5 heures, quelque chose comme ça. C'est pas zéro, mais ça va.
Est-ce que on voit pas la même chose sur YouTube?
Ouais, c'est ça.
There is a delay.
Il y a un
Mais
gros délai.
là,
Mais ça, par contre, il y a un gros délai, c'est normal.
Hmm.
can see this.
Yeah, we should
I don't know.
skip the, I mean, this is Zoom, right? This is Zoom, so
Yes.
yeah,
Attends.
it should be good. This is like,
En real time, hein? Regarde, je crois que t'es dans le passé.
Ouais, j'étais dans le passé. Ah, c'est bon. J'étais dans le passé.
Dans le passé,
Ah ouais, j'étais dans le
Nicolas.
Ouais, Julia, on n'est pas dans le même lien YouTube. Parce qu'en fait, c'est la salle, l'application Zoom qui a créé un nouveau lien.
Ok.
Je ne pouvais pas faire autrement. Donc, du
Hmm.
coup, j'ai mis sur l'ancien lien un lien vers le nouveau lien.
Ok, du coup, bah, l'essentiel, c'est que Venkatesh et la présentation soient là.
Oui, oui, ça c'est bon. Non, mais c'est pour les gens qui veulent se connecter maintenant. Ils peuvent se connecter
Ah
soit
oui,
au Zoom, parce qu'il y a un Zoom public et en fait, les
oui.
micros sont fermés, donc c'est bon, c'est bien fait. Et ils peuvent aussi se connecter au YouTube, le nouveau que j'ai donné. Et s'il y a des gens qui se connectent sur l'ancien lien YouTube, bah, ils verront le nouveau lien normalement. Bon, voilà.
Ok, bah, moi, je reste vigilante s'il y a des gens chez nous qui ont des
Normalement,
soucis.
ils pourront pas se unmute tout seuls, hein, dans tous les cas.
Ok,
And
On va se connecter. Il a
yes,
signé.
you in a while.
Peut-être que Maria est l'une.
Il doit être arrivé. Il doit arriver.
Hmm.
ouais, on voit donc de la caméra de la salle, on voit Venkatesh et la salle. Donc
Mhm.
Venkatesh est très très petit, mais
Ah,
bon.
Ah,
I don't know if you
Tu
can-
peux mettre ta caméra aussi, Venkatesh,
you can-
sur ta ton ordi.
un peu, I think it's on the panel. On the panel, maybe not on the computer. I'm not sure.
Ah, mais ce que je parlais, c'était plus sur son ordi, Venkatesh.
Ah oui.
Il peut mettre la caméra, comme ça on aura aussi ta
Ouais,
vue.
c'est
la
ça,
caméra,
c'est une
ça
bonne
vient
idée.
d'où? Ici et ici, non?
Ah, mais sur ton ordi à toi,
On your computer, you can also put
tu
the
peux
camera.
allumer la caméra.
Ah oui.
Comme ça on aura aussi la vue.
Exactement. Pour les gens qui sont à distance, c'est sympa.
Bah ouais.
Ah, there you go.
Oh,
Voilà,
great.
parfait.
Ok, all right.
Je vais pas pouvoir voir. Ok.
Ouais, c'est en PLS,
Ouais, bientôt, bientôt. C'est pas grave.
on fera après
Fait en-
l'une à la rescousse.
Yes. On va voir si tu m'entends comment ça. Hi, Céline. Céline just
Hi.
arrived.
Hi,
Hi,
how are you?
Venkatesh. So you have to manually ask, put people as having the right to unmute or something like that, or?
I don't know.
Okay, so apparently not.
Yeah.
Apparently not. Okay, so let's just hope that nobody doesn't switch. It's okay, just leave it like this.
Avancer, maybe we need to figure it out, but,
If you go to avancer, advance,
mais c'est pas plutôt participant?
oh,
Yeah.
check.
Même demander enregistrement, demander à l'autre,
Démarrer
demander enregistrement.
Après, c'est, ok, so.
Oh no, nothing like this. Réactiver eux-mêmes leur son, oui.
Ah ouais,
Yes.
réactiver.
Ah no, yeah, you can actually, if you turn that off, people can't.
Ouais,
Tu vois what I mean?
c'est ça, ouais.
De démarrer
Et
le
après,
No, no, leave all that.
dans la vidéo, c'est bon.
Ok.
Par contre, alors, retourne. Ouais,
Autoriser tous les participants.
avancer, tu voir. Peut-être que par défaut, tu peux dire. Non.
Okay, maybe you have to go manually on each participant to allow people. So you have to
Ah, c'est au
allow.
moment de la création de.
No, I guess if you go in the participant list.
Uh-huh.
No, the list,
Ah,
not
list.
the options, not the options, the list. Yes.
Yeah. Ouais, c'est bon.
There you go. So you can allow.
Activer tous les sons en bas.
Okay, you can do that. Oui. And then you have to.
Réactiver Céline
Yeah.
et Julia.
That's it. Demander la réactivation.
Ça par défaut.
Okay, c'est bon. Ouais, Guillaume aussi.
Ouais,
Ouais,
moi,
c'est
ça
bon. Okay, so normally you can turn on and off yourself, Julia and Celine now.
Yes.
Okay.
Cool. Perfect. So we have to do that for everyone. So there's no
Yeah.
bad surprise.
Yeah.
And you can't turn it on again?
Ah.
It's only once.
Ah, ok.
Ouais, c'est genre. Ils peuvent se réactiver, mais s'ils se désactivent, ils peuvent pas faire.
Ah,
It's one shot.
ouais.
Juliet, try now. Should be good.
Yes, it's okay. Now it's okay.
Yeah, okay.
But everyone can.
Okay.
No?
Yeah.
Okay.
But now everyone.
I would just be careful.
We just need to not be photobomb.
Yeah. I guess since there is like a YouTube link, I hope people use that more.
Yes.
Than
No, the only place I shared the Zoom is Discord.
Yeah.
Brain.
Yeah.
It's not a. It is a public channel, but our Discord cannot be joined. Like this.
Yeah.
Because it's not on internet. So
Yeah, it should be good.
should be fine. And from Udemy, I don't know if it's been shared by email or something.
IBM?
Yeah.
No.
There's
It's on us.
There's no official announcement.
Non,
Okay.
pas pour le Zoom. Pour la salle, oui.
Okay. So we should be good.
Ok, bon, on est 10 heures.
How many are we in the jury? How long do we need to finish?
And then,
Next time a little bit. Okay.
ouais, c'est ça, parce qu'on a.
So you
On est
said,
nombreux, hein.
we said, I mean, we, hey, hi
Enzo.
Enzo. Hello.
Hello.
How are you?
Hey Julia.
Hi.
Okay, so now just Karim and
Yeah.
Lynn are missing. Maybe we can talk about that a bit. I don't know.
About the organization?
Yeah.
Constraints
and stuff like that. If people need to leave at certain.
Yeah, so for those who join, like, do you have any constraints? So we have the meeting for like until 2
Yeah.
year,
Well,
but it's supposed to be more like 3 hours usually
in total, yeah.
in total. So we'll be mindful about the time. We have a big jury, a lot of people. So maybe what we can do is like the first round of question with 2-3 question max, especially for the external rapporteur who have in-depth questions, and depending on the time, do a second round
if possible.
if it's possible. But I'm mindful of the time, and I don't want to get over the 3 hours. Yeah.
Perfect.
Okay.
So Maria,
Ouais,
ouais, faudrait
vous avez
peut-être
ton- Si, je mets quand même.
tout le monde est là.
Venkatesh.
How long is your presentation?
45.
45, that's good.
45, yeah.
Mhm.
Karine.
Okay. Il n'y a plus que l'une.
Que l'une, ouais.
Elle a dit qu'elle serait dans 10 heures. Ah ouais.
Almost there.
Okay.
Deuxième étage, oui. Non. Si,
Deuxième.
si, on dit deuxième étage.
Ouais, ça, je ne connais pas.
The whole line.
Cheers.
Pour l'ordre des questions. Ouais. Alors, Céline, voilà.
Ah non, lui. peut-être.
Alors, nous, président.
Non,
Non,
mais
présidente.
l'une est
Elle est
représentante du doyen. Ah, normalement,
Oui, normalement,
on n'a pas de question.
elle a pas besoin, mais dans ce cas-là, c'est le même. Pour elle,
C'est intéressant. Ouais.
Là, tu peux dire que.
Et après, nous,
Moi, j'en parle aussi, non?
Ah oui.
Ouais.
Avant, non. Moi, je...
On va terminer à 5
Non,
Non, justement.
Non, on verra. Oui, je vais avoir parlé de la Chine.
Voilà, moi, voilà, ça.
Ouais, c'est vrai.
Juste
Pour.
Last but not least. Hello, Lise. Okay. Hello.
Du coup, c'est vrai qu'on est 8.
Ouais. Désolé.
Okay, good. So since we are all there now, 8 people, it's like a big jury. So we're gonna start with the presentation of Venkatesh for 45 minutes. And then we're gonna move to the questions. And as I said, like I will try to be mindful so that we're gonna stick to the 3 hours we have been allocated. So Venkatesh, the floor is yours.
So thank you, Mr. President, for the introduction, and thank you to the jury members for accepting to be part of the jury. Oh, sorry. Sorry. Yeah, good. Perfect. Okay. So without further ado, let's get started. So I've been very excited to be able to show you what I've been working on for the past 4 years, entitled Temporal, Spectral, and Structure Function Signatures of Brain Dynamics Across States of Consciousness. So this work is carried out under the framework of co-tutel, so co-supervised PhD program between two universities, IMT Atlantic in France, supervised by Nicolas and Julia, and Université Montréal, supervised by Karim. So one could loosely define consciousness as a spectrum indexed by experiential richness. Think of it like richness in the experience. Anesthesia is a state where there is like restricted, minimally rich experience as compared to more vivid ones, such as dreaming. And the normal wakeful state encompasses the states with the full range of day-to-day experiences, such as perception, emotion, and the sense of self. So even within that, there are like ongoing stimuli, such as naturalistic movie watching, that elicits a richer brain dynamics. Psychedelics push the brain towards even fuller, richer brain dynamics, such as hallucinations and dissolution or disruption in the sense of self. So in this sense, anesthesia reflects the state with restricted repertoire of brain function, brain state rather, and psychedelics produce a more broadened, enhanced repertoire of brain state. So how do we? So the primary question that we are trying to address in this thesis is as follows. So how are electrophysiological brain dynamics structured across a rich naturalistic stimulus and rich altered state, such as psychedelic-induced altered states? So how do we study this? So essentially, it's through neuroscience. So populations of precise synchrony between populations of neurons gives rise to what we call as activity oscillations in the temporal domain. So what do we mean by oscillations here? Let's get concrete. So populations of neurons organized in a parallel manner tangential to the surface mostly gives rise to what we call as activity, as represented here. These are like synchronized activity oscillating at a particular frequency. Prior to the arrow here, the activity is less synchronized when the subjects have eyes open as compared to more synchronized activity when the eyes have been closed. So these oscillatory patterns could be extended towards other conscious states, such as low frequency dominant, coma, higher frequency dominant, drowsiness, states of relaxment, and the states of excitement. So turns out that there is more to the activity recorded than just the synchronous activity. There is also asynchronous background activity that contributes to the activity that record. So this can be characterized by the aperiodic component, aperiodic exponent here. So there is a, if you look at this, there is a linear drop across frequency range, and the total power represents a superimposition of not just the periodic background activity, but also the oscillatory activity that sits on top of the asynchronous activity. So we have this. How do we deal with this? So consider you have the signal, and we compute the power spectral density. We parameterize the spectrum into the constituting components to isolate the arrhythmic component and the rhythmic component. This enables us to understand in an unambiguous manner where the source of the effect lies. So it turns out that this 1/f arrhythmic component indexes the richness of the brain dynamics. So consider this power spectrum here. It's a steepened power spectrum. So which means that there is a low frequency dominant activity there. And this corresponds to states like coma. And and there is a shift in the peak, for instance, shifts like here. So what it means is that the power shifts towards higher frequency range could index states such as states of excitement. So there are many ways to quantify the richness of the brain dynamics. Two of them include Lempel-Ziv complexity and Higuchi fractal dimension. Let's go through Lempel-Ziv complexity first of all. So you have the continuous signal. You can binarize using mean or median. Put 1 if it is higher than the threshold, 0 if it's lower. Get the binary string. Lempel-Ziv complexity essentially quantifies the compressibility, the predictability of the signal. Moving towards Higuchi fractal dimension, intuitively speaking, you can consider as a yardstick of varying temporal scale, starting with the quartz one, and Higuchi fractal dimension essentially attempts to trace the signal, the activity there. So to be more precise, it's the relationship between the varying temporal scales and the traceability of the signal indexed by the slope of the relationship between the two. So these tools have been used, and to reveal more about the temporal organization, let's go through what we know so far. 1 over f, the aperiodic slope, has been shown to reflect the balance between excitation and inhibition ratio, and the same has been shown to track the intensity of the Lempel-Ziv complexity. And psychedelics are known to modulate both the oscillatory part of the signal, but also the aperiodic asynchronous activity. Here you're looking at the total power that constitutes both the periodic and the aperiodic component. And using the tools that we described earlier, we can parameterize, isolate the arrhythmic component. And if you can look at it, so there is a flattening of the slope there. And we can isolate the rhythmic part of the activity. And this reveals that there is an oscillatory weakening. So to sum up, psychedelics disrupt oscillatory rhythms and increase neural complexity. In fact, there is more to it than just temporal dynamics alone, because the regions do not exist in isolation. They are interconnected, talking to one another, defined by the spatial propagation of the activity. So what do we mean by spatial organization here? So these studies have revealed that unimodal cortices, such as sensory motor regions, they anchor on one end of the spectrum as compared to the multimodal transmodal cortices encompassing DMN related networks. So you could think of it like it's indexed by specialization, in a sense that unimodal is functionally specialized to one modality, as compared to transmodal cortices are more associative in nature. So also you could think of it like the source of the effect it's processing, in a sense that sensory regions are lean towards the outer signal, as compared to DMN, the transmodal cortices, turning towards more inward spontaneous activity. So this shows that there is a gradient spanning between visual and the DMN regions. So other phenomena of the spatial organization is the structure function coupling. So studies have used structural connectivity and functional connectivity matrices and found that there is a monotonic relationship between the two. It's great. Can we get, so this is like a comparison between the structural connectivity and functional connectivity matrices. Can we analyze in a more direct manner? It turns out, yes. Let's go through, let me walk you through in detail. So you have the functional activity record at the scalp level, go back to the source space. On the other hand, you have the diffusion scans, tractography, construct the anatomical connectome. You take the signal, put it on top of the graph, thanks to graph signal processing, and see the spatial propagation of the activity. So graph signal processing enables a multimodal fusion between the structural data and the functional data. So far we quantified the spatial propagation of the activity. How do we characterize the structure function coupling? So you have the diffusion scans, tractography, and decompose its corresponding harmonics. They are orthonormal, meaning that they are orthogonal. They form the complete basis set. So you can represent any activity as a weighted linear combination as depicted here. Each of the harmonics carries a specific meaning. The low frequency harmonics tend to be brain wide or like long wavelength long wavelength harmonics. They capture the activity that spans across that utilizes the anatomical network termed as coupled to the structure. On the other end, you have the high frequency harmonics that capture the activity that's more localized, meaning decoupled from the structure. So we used structural decoupling index to characterize structure function coupling. So essentially, it quantifies the ratio between decoupled activity and the coupled activity. So these tools the connectome harmonics have been used to reveal more about the spatial organization. So let's go through one at a time. It turns out that EEG activity during event-related potential, meaning the activity is locked to the particular stimulus, can be compactly represented using connectome harmonics, meaning that a subset of the connectome harmonics would suffice to represent the EEG activity. And studies have used SDI, structural decoupling index, to quantify the structure function relationship. They found a similar organization spanning between unimodal codices anchor on the coupling end towards transmodal codices decoupled on the other end. Psychedelics, the effects of psychedelics on structure function coupling have been analyzed with fMRI signal, and they found that there is a shift in the graph spectral power from low frequency harmonics towards high frequency harmonics, suggesting a more modular organization of of fMRI activity. To sum up, EEG activity can be compactly represented. EEG activity during ERP can be compactly represented, and the psychedelics induce a more modular fMRI signal. So, in this thesis, we embrace the spatiotemporality. Spatiotemporality and analyze the both aspects in a joint manner. So let's formulate the questions here. So structure function coupling has been analyzed with electrophysiology with a narrower focus such as ERP. However, it is not fully representative of the day-to-day life. Naturalistic stimulus such as movie watching could elicit a different dynamics, and how is the structure function organized for those conditions is not yet well characterized. And structure function coupling has been analyzed only with the fMRI signal, and however the psychedelics have been shown to modulate the neurovascular coupling. So whatever changes you observe include not just the drug-induced effect, but also the vascular confounds. So more direct electrophysiological dynamics would provide unambiguous evidence. On the temporal side, LSD has been shown to disrupt both the oscillatory rhythms, but also shifts the spectral peaks. So whether there is actually a genuine desynchronization is not yet characterized. And numerous studies have analyzed the complexity under electrophysiology, but it remains not yet fully characterized. So we formulate 3 questions surrounding the outlined questions there. Let's go through one of them, one at a time. So how does the structure function coupling organization manifest during naturalistic stimulus? That's tackled in study 1. Study 2 tackles how does LST modulate the temporal and spectral dynamics. And the third study tackles how does LST induce reorganization in the structure function coupling. So here is a bird's eye view of the methods. There are 6 different modules here, starting with the pre-processing and the source localization of EEG and the MEG signal. That's on the functional side. And the connector on the structural side, we derive structural connectivity matrices for the individual subjects for study 1 and derive structural connectivity matrices for HCP subjects for study 3 and decompose their corresponding harmonics and quantify the complexity and parameterize the spectrum using Lempel-Sieve complexity, Higuchi fractal dimension, and spectra. So let's get into each one of the studies here. So a little bit of a background, structure function coupling has been analyzed with fMRI and with electrophysiology with a narrower focus, ERP and resting state. However, natural physics stimulus elicits a particular brain dynamics, and how is the structure function coupling organized or reorganized is not yet well characterized. So we aim to probe this question and set up the aim. How to establish how structural connectivity constrains the large-scale dynamics during naturalistic movie watching and compare against resting state. And we formulate 2 research questions here surrounding the aim. First one being, how is the continuous EEG constrained by the structural connectivity, and whether this is consistent over time is the next question. So here is the study-specific material. We utilized healthy brain network involving 40+ subjects under resting state and the movie watching. EEG was recorded, and we perform source localization using E-Loretta inverse modeling, and we apply on top HCP MMP atlas to extract the activity from 360+ regions. And this on the functional activity side. On the connectome side, we derive subject-specific structural connectivity matrices, decompose its harmonics, functional activity, structural connectome harmonics, map it to quantify structure function coupling using structural decoupling index across various frequency range, starting with theta, alpha, beta, gamma, and on top of that, wideband. So let's get into the results here. So you're looking at the group level spatial maps. Blue end corresponds to coupling and the red end corresponds to decoupling. So as we can see here, there is a brain-wide coupling and the focal decoupling patterns there. So to interpret it further, we perform what we call meta-analysis. Essentially, we take the spatial map, bin it at 10 percentile increments that corresponds to the x-axis there, and look into the database of literature to see which topic terms, y-axis, describe the corresponding spatial maps. So as you can see here, visual codices, systems such as visual codices, motor codices, and the auditory unimodal systems, they anchor on the coupling side as opposed to more transmodal systems such as social function systems implicated in social, emotion, mental imagery, and so on. They span across coupling to decoupling axis, indicating a weaker coupling. This pattern is quite consistent also with the resting state. And the unimodal systems anchor on the coupling side, and the transmodal systems coupling to decoupling, indicating a weaker coupling there. So is it consistent over time? So that's the question we address here using standard deviation as the index, and on top of that we threshold the distribution. Bottom 25 percentile corresponds. These are the regions corresponding to the regions with the minimal temporal fluctuations, and on the opposite end, regions with the highest temporal fluctuations. So unimodal systems, and in addition to that, PCC, they exhibit a minimal temporal fluctuation there. First row, and the second row corresponds to transmodal regions, exhibiting a more dynamic fluctuations. So the takeaways are as follows. Unimodal systems, they are anchored, they are coupled, they are strongly coupled to the structure, and the transmodal codices exhibit a weaker coupling. And the second takeaway being that the unimodal systems, they are stable over the entire video as compared to association codices exhibiting a dynamic fluctuation. The overall finding is that EG dynamics are constrained by the connectome in a spatially and temporally specific manner. So far we established the coupling organization under a rich normal wakeful state. And how is the brain dynamics reorganized during a rich altered state is our next question. So here's a familiar spectrum. So you have the psychedelics, and they induce changes in the neurophysiology, in this context, temporal aspect, and that would in turn produce what's called the enhanced, broadened repertoire. How do the changes in temporal reorganization manifest is our focus in this study. So a little bit of a background. LSD induces both the desynchronization and alongside the peak shift. Consider this scenario. So you have the two power spectrum here. So there is a systematic power shift without any weakening of power. If you do area under the curve, it would manifest as a massive desynchronization. So this calls for caution to analyze the desynchronization after correcting for such shifts. And the aim of this study is to probe the spatial specificity of the LSD-induced peak shifts and investigate the desynchronization after accounting for such peak shifts. So we formulate two hypotheses surrounding this aim. First one being that there is a region-specific, there's a spatial specificity to the peak shifts, and there is actually a genuine oscillatory power weakening after controlling for such peak shifts. So here is a study-specific material. We used the dataset from Kahat-Harris involving 17 subjects in a single-blind crossover design. Subjects took LSD or either LSD or placebo, and MEG was recorded under resting state with eyes closed, resting state without eyes closed. We performed source localization using DSPM inverse modeling, and as before, we apply personalization on top to extract the activity from 360 regions. And we quantify the temporal complexity using impulsive complexity and the Higuchi fractal dimension, and we parameterize the spectrum using SpecParam. So let's get into the results, starting with this one. So here you're looking at the global region averaged power spectrum. On top, you're looking at the total power. Bottom, you're looking at only the oscillatory activity. Two things stand out. One, there is a significant desynchronization after correcting for 1 over F, and there is a shift in the oscillatory peaks in alpha and beta range. This is a global region average spectrum. Whether this holds true for the regions, it turns out yes. So for alpha, there is a brain-wide shift in the peaks, shifting up to 2 Hertz, and for beta, it's more localized. However, it's shifting up to 5 Hertz. So as alluded earlier, LSD modulates. So after accounting for the peak shifts, do we see still a genuine? Do we still see a desynchronization? Is our next question. It turns out yes. The first row corresponds to changes in the spectral power within alpha range, and for the second one, we take peak shifts into account. We track the spectral peaks and quantify the change at the peak, and we find a desynchronization, suggesting that there is a genuine desynchronization. As alluded earlier, the LSD modulates not just the oscillatory part of the signal, but also the asynchronous aperiodic activity. And let's look at the changes here. There is a brain-wide change in the exponent, indicating that flattening of the slope it's a brain-wide effect. And this LSD also increases the fractal dimensionality brain-wide. So the LSD wouldn't be modulating the entire brain in the same way. To understand the gradient, we perform meta-analysis here, perform the meta-analysis for all the features. Here I'm showing you for the subset of it, starting with the alpha peak power. The blue, the left end of the x-axis corresponds to the systems with maximal drug effect, strongest attenuation, and the right end of the x-axis corresponds to minimal drug effect. Systems implicated for auditory processing, emotion, and language-related ones, they exhibit a maximal drug effect as opposed to motor system exhibiting a minimal drug effect. This pattern is quite consistent also with the 1/f slope. Sensory systems, maximal drug effect; motor systems, minimal drug effect. To sum up, LSD disrupts the sensory and the language-related functions the most, and while sparing the motor cortices. So the takeaways are as follows. So LSD shifts the peak frequencies upward in a region-specific manner, and after accounting for those peak shifts, we have evidence to suggest that there is a genuine oscillatory attenuation, especially in the alpha and beta range. So the overall finding is that LSD reconfigures brain dynamics through oscillatory suppression, peak shifts, and increases the fractal dimensionality. So far, we characterized the LSD's perturbations on functional dynamics, and how about the perturbations on the link between structure and function is our next question. So here's again the familiar spectrum. Psychedelics, when subjects take psychedelics, there would be changes in the neurophysiology. In this context, spatial reorganization that would in turn produce what we refer to as enhanced, broadened repertoire of brain state. And how do the changes manifest is our next question that we address in this study. So a little bit of a background here. Structure function coupling reorganization has been characterized with fMRI signal. However, they are indirect metabolic measure, and psychedelics have been shown to modulate the neurovascular coupling. What it means is that what we observe as changes in the BOLD could reflect not just the drug-induced changes, but also the superimposition of the vascular confounds. So more direct electrophysiological dynamics would provide a more unambiguous answer. So we formulate the hypothesis surrounding this aim, stated as follows. LSD selectively relaxes constraints on slow integrative rhythms. So we used the functional activity from the previous study. On the connectome side, we derived the structural connectivity matrices for 1,000+ HCP subjects and parcel it on the HCP atlas, same as the functional activity, and decompose its corresponding harmonics, functional activity, structural connectome harmonics, put two of them together to quantify structure function coupling using structural decoupling index across various frequency bands, starting with the theta, alpha, beta, and gamma bands. Let's get into the results here. What you're going to see is the contrast map. Blue end corresponds to LSD strengthening the coupling, and the red end corresponds to LSD strengthening decoupling. So what we are observing here is that LSD is strengthening the decoupling for the slower rhythms, as opposed to a more heterogeneous effect in the high frequency rhythms, encompassing both the strengthened coupling and the strengthened decoupling. So we perform meta-analysis here for all the frequency bands. Here I'm showing you the alpha band for the interest of time. The blue end of the axis, the left end of the axis corresponds to strengthened coupling, and the right end of the x-axis corresponds to strengthened decoupling. So systems, auditory systems, they are anchored on the strengthened coupling side, and attention systems, attention systems and visual systems, they exhibit a more strengthened decoupling. So the takeaways are as follows. LSD decouples the slower rhythms from the structure, and there is a preferential targeting the visual attentional systems on top of the auditory systems. So the overall finding is that LSD relaxes structural constraints in a frequency and network-specific manner. So far we looked at individual studies. Let's put all of them together. And first of all, to recap, study 1 revealed that LSD structure function coupling is hierarchically organized during normal wakeful state. And the study 2 revealed LSD retunes slower rhythms, flattens the slope, and increases fractal dimensionality. And the study 3 revealed that, as we have just seen, LSD relaxes structural constraints in a frequency and network-specific manner. Overall, this thesis reveals that electrophysiological dynamics are hierarchically constrained by the structure, and the LSD reconfigures by retuning oscillations, increasing complexity, and decoupling the slow rhythms from anatomy. So how does it pan out with the existing framework? So let's talk about the gradient organization here. We have the following evidence. Electrophysiological structure function coupling is organized along unimodal to transmodal cortices. This speaks to the claim of structure function coupling is hierarchically organized across unimodal and transmodal cortices, and the evidence of LSD decoupling specifically in a targeted manner, visual and attentional systems, speaks to the point of reorganization in a system-specific manner, arguing against the universal disruption widely reported in the previous literature. Despite its limitations, entropic brain hypothesis provides a simple framework to interpret our findings, and let's look at each one of those. So we have the following evidence. Decoupling of the functional activity from the structure, desynchronization of alpha and beta rhythms, increased fractal dimensionality and the complexity, and flattening of the slope. These all evidence put together speak to the point of LSD nudging the brain towards a more dynamic, flexible state, potentially contributing to the broadened functional repertoire. So this thesis could be extended in several different ways, and I'm mentioning like a few of the most noteworthy things, starting with the translational scope. So psychedelic literature usually concerns with two aspects. One of them, changes in the neurophysiology, and the other one is investigating the potential of psychedelics in the clinical context. There is a strong disconnect between the two, meaning that how do these changes in neurophysiology underpin the clinical outcomes? One of the things I'm most excited about is to bridge the psychedelic mechanism of action with the clinical benefit. And the second perspective being the inferential scope. So the observed effects, what we call as observed effects, include not just a drug-induced effect, but also include a superimposition of the expectancy effects from the subjects. So one way to go about it is to explicitly ask the subject about the expectancy and consider this as a covariate and include in the statistical modeling to understand the drug-induced effect in a more explicit, unambiguous manner. And the third one, the final one, being the framework scope. We quantify the structure function coupling for the signal that has both the arrhythmic and the rhythmic component. It's not at the given that they both behave with the structure in the similar way. So explicit characterization would be essential in this context. So let's zoom out and see what we can take away from the thesis. So starting with the brain work. So the brain's function is organized across 3 dimensions here: spatial, spectral, and temporal axis. And the LSD coherently reshapes the brain dynamics across these said dimensions by decoupling structure, by enriching temporal complexity, by shifting the spectral energy to faster rhythms. More broadly, as consciousness in general, altered conscious state under LSD reflect the structure and a multidimensional organization of brain dynamics. The takeaway of the thesis is as follows. LSD produces an altered conscious state by broadening the dynamic repertoire across space, time, and frequency. With that, okay, there's one more slide. So this thesis had contributed to 3 papers. Study 1 published at Imaging Neuroscience. Study 2 is under review at under revision at Communications Biology, and the third study is under review at Network Neuroscience. I've got opportunities to present these works as talks in local and international conferences, including ASSE, and opportunities to present as posters in local and international conferences, including OHPM. With that said, this work is not possible without the massive help of the labs with amazing collaborators and a bunch of open source libraries, and with massive thanks to the funding agencies as well. With that concludes my presentation, and happy to take questions.
Thank you so much, Venkatesh. So we're gonna enter the phase for the questions, and we're gonna start with Selina Villente, who was rapporteur.
Okay. Well, thank you first for this nice presentation. So my first question is very more general about your framework and what you actually presented at the beginning of your presentation, where you were talking about the temporal organization, spatial organization, and the spectral aspect of brain activity. From what I'm seeing is that all of the metrics and methods that you presented for quantifying temporal organization is really at the univariate level. What I mean by that is that you're looking at each signal and its spectrum distribution at each channel level. So what can you say about multivariate organization of the brain? So looking at simultaneously spatial and temporal interactions. If it's not clear, I can clarify further.
No, it is clear. Thank you so much.
Okay.
Yeah, thanks for the question. So indeed, temporal dynamics, when you look at it, it's indeed looking at only one of the regions. And obviously there is a spatiotemporality, meaning that we cannot dissociate one with the other, meaning like one cannot exist without the other. So one of the ways to address this question probably would be spectral aspect is like the common denominator between the time and space, in a sense that they represent large scale or local activity. So in terms of like structure function coupling, for instance, studies from Patrick Hegman, for instance, in 2023, they looked at, in a joint manner, the structure function reorganization across time and the spectrum, for instance, meaning that what's the contribution of connectome harmonics across time and space. They call it like a joint vertex analysis, I believe. So that would be a way to go about it. We started this question in like 2021. So this was the focus for the study one, to quantify the structure function coupling in a rather independent manner. And we performed similar analysis with psychedelics to be able to compare between the two studies.
Right. So I'm going to get a bit more specific, if it's okay, about this question. So if you actually look at your chapter 4 and you look at temporal organization metrics and you talk about Lempel-Ziff and Higuchi fractal dimension, those are all univariate measures. So when I say multivariate, I'm talking about interactions between different regions. So do you have a sense of how you would extend, for example, the work in chapter 4 into a multivariate setting where you would actually look at the organization of the brain rather than looking at each region individually?
I have to say that's a great question, and I haven't thought about it deep enough to be able to operationalize in practice.
Okay.
Oh yeah, okay, yes, yeah. So basically, thank you, Karim. So functional connectivity could be a way. For instance, in fact, I've been working on the functional connectivity for like over a couple of months. So that could, for instance, like oscillatory coupling could include both aspects at the same time. And they have some reasons for that as well, actually. So basically, so the study 4 shows that there is a shift in the spectral peaks, right? So this is like a first study to this is the way to establish there is a shift in the peaks that would require more thought as compared to canonical analysis, and this is a starting point, and many studies could follow suit in a sense that anywhere that requires oscillatory coupling, for instance, would require careful rethought. So this was our primary focus here. So what we performed here is to track the peak frequency wherever it's shifting, and then compute the functional connectivity there. So the idea is that the hypothesis rather is that it would reflect when you're tracking the peak frequency shifts, it would reflect a genuine either desynchronization or the resynchronization reflected in the functional connectivity measures. So for that, we considered different frequency functional connectivity metrics, WPLI phase lag based ones, coherence based one, and amplitude based ones. So what we found is that so here the canonical represents, we do not touch, we consider the functional connectivity as is. We do not shift both of them. We do not consider the shift at all. That reflects the standard analysis, and adaptive reflects you track the peak frequency shift, and we consider that machine learning analysis would classify, would discriminate better when there is adaptive frequency range. And we do not have clear evidence pointing that the adaptive, when you're comparing the frequency, when you're tracking the frequency range, it is more discriminative than the other. So long story short, the functional connectivity could be a way to analyze both the space and time in a more multivariate fashion.
Okay, yeah, that's exactly what I was going with. So thank you for
Thank you so much.
yeah, thanks for sharing the slides. So what is your conclusion then? With LSD, what happens to functional connectivity in the alpha band?
Sorry, could you repeat the last part of the question?
Yeah, so what happens to functional connectivity in the alpha band with LSD versus placebo?
So this is in the early stage actually. So this is like a preliminary results. So there are different things. I could get into details, but for the interest of time, so here it's not yet conclusive that adapting frequency range provides like a more discriminatory power between the two, and this could be related to like different regions, different reasons rather, such as like you have the functional connectivity matrices here. This is like at the edge level analysis, and we perform like a seed-based and network-based analysis as well. One issue or one caveat, one downside with edge-based edge level analysis is that there is like a lot of statistical comparisons. You have to correct for the multiple comparison corrections here, and with the limited sample size, it gets like more complicated to be able to conclusively show that there is the adaptive frequency range is like better than the other. We are trying we are exploring a bunch of different approaches, and we are looking forward to the results on this.
Okay, thank you. So should I continue or just
Yes, please.
okay. So my next question is much more specific to the structural functional coupling that you looked at. I think it was chapter 5, which is the last one with LSD. And you took the connectome as a template from a human connectome project. So how valid is that assumption of using a template connectome to do the structural functional coupling analysis?
Yeah, thank you for the question. So indeed, we used template connectome from HCP subjects. The important thing that to note is that this could completely suppress the idiosyncrasy associated with each individual subjects, and whatever the bias it could have, it could have like biases because we used structural connectivity matrices for the LSD condition and placebo condition. Thankfully, the effects are mitigated by the within subject design. The bias would be equivalent between both conditions there. So to sum up, the idiosyncrasy is not taken into account, but the bias would be spread out between both conditions there.
Okay, yeah, I get your point about that. So is that representative of the population that you used in your study? Like at least is the age group matching?
Yeah, great question. So yeah, so basically, as I recall, the subject group, the demographics of the LSD data set, I believe is between 30 to 40, I think, if I recall correctly. And HCP is young adults, and we are banking on the fact that there is 1,000+ subjects that could be reflective of the population structural connectivity dynamics there.
There is time for one question, Sin.
Okay, sure. So the my other question is also related to that chapter, the same one which you look at LSD-induced modulations on structure-function coupling, which we kind of touched upon this before. So one thing that I noticed in this chapter is that there is no discussion on the temporal evolution of LSD's effects relative to administration time. So even if you don't have a complete dynamic analysis of structural functional connectivity, do you have any idea if there are any significant changes between early and late time points following the exposure to LSD in terms of this SDI metric, maybe?
Yeah, thank you for the question. So indeed, you're right. We focused on the time aspect with the study 1, but that one is like missing in the study 3. There are some several reasons associated to it, and then the crucial thing here being that if you look at the pharmacokinetics of LSD, so it lasts the high period, so to speak, the drug effects, they last up to like 10 to 12 hours. You're looking at some of the results here. You can ignore the okay, you can consider all the conditions here. So basically one could do one of the things that we couldn't do but we wanted to is related to the fact that LSD condition was quite noisy, that resulted in more discontinuous signal. So this was like not consistent across subjects, that essentially precluded doing time varying structural structure function coupling there. So one way to go about it, as you suggest, could be to consider the last and the first time points. So what I am thinking is that because the if you look at the pharmacokinetics of LSD lasts like much longer, like it's in hours, and the data was collected 4 hours after the ingestion. That's like after, if you look at the plots here, that's a little bit after the peak drug effect, and the MEG signal is like about 7 minutes long. So if you look at if you compare the pharmacokinetics between the 7 minutes, there wouldn't be technically a massive difference in terms of, at least theoretically, on the drug side. But as an exploratory analysis, this could be absolutely a way to see what's going on in terms of time resolved structure function characterization there.
Okay. Thank you.
Great. Thank you. Now we're gonna pass to Enzo Tagliasucci for the other rapporteur.
Okay, thank you very much. Thank you very much for the presentation. I really like the several things about this thesis. So I like the focus on the electrophysiology aspect of the LSD experience or the psychedelic state more generally, because as you said, we don't know a lot about this. Most of the studies are based on fMRI, and there are lots of potential issues with the fMRI signal under psychedelics. And I also like very much the careful approach of trying to distinguish the oscillatory component from the 1 over F component of the signals. So my questions are, I have 3 questions. Some of them will be subdivided into smaller sub-questions, but the first one is something that I also wrote in the report, that because your method to compute the structural functional coupling is quite sophisticated, so you are not simply computing all the pairways connectivity strengths, for example, and comparing those matrices. So what you're doing is a bit more convoluted. After reading your thesis, I don't get a clear idea. At least I don't do it, because maybe I don't understand the methods well enough. But I don't have a clear idea of how strong this coupling is, because in other words, what I think could be useful is to have some comparison with a more straightforward method to try to understand the intensity of this coupling. For example, is it true that the regions with stronger structural coupling are those that have stronger coupling, functional coupling in different frequency bands, and so on and so forth? Which to answer this question, maybe just looking at the pairwise couplings in both modalities and comparing them, even if it's not as sophisticated as what you did, could be helpful. So my question is whether you looked into this, or maybe what's happening is that this is implicit already in the analysis you did. I'm not seeing that. So maybe if you could help me understand that then.
Yeah, thank you for the question. So from one of the answers to Celine, we indeed performed kind of like after the fact. We, as an extension to finding the spectral peaks, we looked into the oscillatory coupling, meaning indexed by basically to see the functional connectivity there. So here are some screenshot of the results there. So we looked at three aspects: the main effect drug, main effect shifting the canonical, the frequency definition, and the interaction between the two. What's the relationship? What kind of effects do we have if we shift with or without? So we found a significant difference on the band definition, and not significant effect for the drug and the interaction between the two. And we consider different metrics: phase lag based, and coherence based, and amplitude based ones. As mentioned earlier, the downsides of doing this analysis is related to statistical comparisons, because you can obviously look at the functional connectivity in different manner, age level, seed based, motivated by the previous literature. We have like results as well, I will show you as well. So, and then the another thing could be like network based. So this is like in the early stages, preliminary results, and we so here to elaborate a little bit, the key takeaway of this result figure is that we do not find conclusive evidence suggesting that adaptive frequency would produce a more unambiguous characterization of the functional coupling or decoupling. So this directly complements the structure function coupling analysis. So there you're looking at the amplitude of the signal propagated through the structural connectivity. Here you're looking at the coupling of the coupling at the activity level.
Yeah. But this is the analysis of the functional coupling, right? Or
yeah.
Okay. Yeah, but what I meant is, just looking at the because you start with a matrix of structural connectivity at some point, right? Which is the DTI matrix. So you can basically plot that matrix, and you can also plot these pairwise connectivity matrices in the functional sense. So if you do that, you can simply look, with your own eyes how similar they are. Of course, then you introduce some metrics to quantify that. But my point is that, in the fMRI literature, if you look at the early papers that try to understand if there was correspondence between the structural and functional connectivity in the brain, they work with that object, the matrices, and then just, start visualizing them and computing the correlation and so on and so forth between those matrices, which is a much more straightforward way to do things. But again, it has the advantage of, because other way to put my comment is that we are looking at differences in the structural functional coupling between conditions, and that is fine. but I also want to know, if besides the difference, what is kind of the strength of the coupling that you can discover with your method or with your machine modality you're using. So and maybe compare it, in my own mind with what I know about the structural function coupling estimated with fMRI, for example. And for that, I just wanted to see a more straightforward visualization, even of the things. I mean, so this is something that maybe in a paper will end up being in a supplementary material, but for me, especially since I don't know very well the method, I thought that could be useful, but just as a very, very general comment,
Yeah.
Mhm.
Sorry, go on. Yeah, sorry.
No, please go on, please go on.
Yeah, totally agree with the sentiment.
Mhm.
So you have the functional connectivity there, right? So basically, this is like the functional connectivity matrices we compute with different functional connectivity metrics, phase lag, a bunch of different metrics there. So so you have the functional connectivity, and the complication is that how do we interpret this? There are many ways to go for it. One way would be to use some sort of like graph theory metrics to analyze, I don't know, integration or segregation at the functional level, for instance. That could be a way. The other way we proceeded to do is basically, because they are symmetric matrices, you can flatten the functional connectivity matrices and compare between LST and the placebo conditions to see how LST modulates at the functional level. That's the machine learning classification here, in a sense that you flatten the functional connectivity matrices and see the discrimination between the two. It would indeed, so you can look at the functional connectivity matrices, but there needs to be a more statistical characterization to be able to see what is it that what are the observed effects. If you see what I'm trying to say, yeah.
Okay. Great. So the second question is about so something that you used at several points in your thesis was the neurosynth decoding. So because you wanted to compare the maps that you obtained with the maps linked to different cognitive functions. So I have two comments or questions related to this. So first is that the neurosynth decoding comes mostly, if I'm correct, from fMRI studies, right? So these are maps that are obtained using a different modality compared to the one you used. So my question is, if you think you need to be careful about this mixing of modalities between your results and the source of the maps. And the second is more of an advice, perhaps, or something that maybe you looked into that, but you did not report that. Of all the spatial decoding, if you want to call it that. So the one that I wanted to see the most, and I think that could be the most useful, is the comparison not with the cognitive function, but with the map of receptor affinity of the drugs, because you know that it's very, very well or relatively well understood the molecular action of these compounds. So it's a 2I partial agonism and so on and so forth. And even between different drugs, there could be some variation in the affinity for all the receptors. And there are maps that estimate the distribution of these receptors using PET imaging. And if I'm not wrong, maybe my memory is not is failing me, but I don't recall that you compare the distribution of the receptors to the maps that you obtained in your analysis. And if you did not do that, well, I think it could be useful to do that. So those are my main two comments or questions about this.
Yeah. Thank you for the question. So the map that you're referring to, I believe, is from Beliveau 2017. They quantified for the first time with PET imaging the characterization basically for various receptors, HT1, A, 2A, and so on. We performed coupling. We performed the regression analysis, comparing the changes in the coupling organization with 5-HT2A receptor, and between this changes in the peak shift and so on. We indeed performed that. However, we did not. We find a minuscule correlation, however, not significant between the two. So we did not, because of this, we did not push further on that. This could be linked to the fact that there is like a low sample size with our study. Yeah.
Yeah, and then maybe just to wrap it up, a very short question or comment, which is also related to what Celine mentioned about the use of the group average connectome, which I think that the main problem with that is that you lose the between subject variance, right? So you end up having one map of structural functional coupling for each subject, but since you don't have an individual, or you end up having like the functional estimate of the coupling for each subject, but since you don't have an individual structural connectivity coupling map for each participant, then you can't explain that part of the variance with the group average. So I think that's the problem. And that's just a comment. I mean, that's nothing you can do because people don't tend to measure DTI in this study for some reason. So that, I mean, you can't do anything about that. But the other question I had is that if you thought about also on the topic of inter-individual variance, if you thought about running correlations between your findings and the subjective reports of the experiences, because there is some variance there. Like some subjects report more visual experiences and so on and so forth, or even simpler than that, more intense experiences, some subjects than others. And if you thought about comparing the intensity, for example, of the experience, either general or in specific domains, to the main findings of your neuroimaging analysis.
Yeah. Thank you for the question. So the subjective reports were part of the data set. So we are lucky in that aspect to be able to compare the changes in the neurophysiology with the subjective reports. So it was collected across the phenomenological aspects include imagery aspect, ego dissolution, and mood and emotional arousal and so on. In total, 6 aspects. So we compared the changes between neurophysiology and the subjective reports. However, for both of the studies, we did compare that. So on the structural function side, we found a strong, significant relationship between decoupling in the DMN, meaning that increased decoupling in DMN, particularly PCC, tracked the intensity of ego dissolution. So that's one of the results. And for the rest of the comparisons, we did not find significant correlation between the two. There are some caveats associated with the subjective reports. The way it was collected back in 2014 or something. So it was brought up by the collaborators that it was not done in a rigorous fashion. Also, to add to the fact that the questions that were asked to the subjects were formulated as retrospectively, the question, the subject reports, they were asked to report the current relative experience, relative intensity as compared to their peak drug effect. So this adds complications. This adds a noise to the analysis, potentially dropping down the sensitivity of the regression analysis, and add to the fact that the low sample size, it creates a bit of a complication to push hard on the the regression analysis.
That's the main problem, I think, because you have a noise of not only in the report itself, but also in the capacity of the subjects to introspect this. And I don't think this can be done with this sample size, in a reliable way. So anyways, thank you. Those are all my questions for the moment.
Great, thank you.
Thank you so much, Enzo. So we're gonna move to Maria Giulia Preti.
Yes. Hi, and thanks a lot for your presentation. Very clear, very interesting. Also, I really enjoyed reading your manuscript and the different works. So I will start from a question that concerns maybe the first, the first work. So I think chapter 3. And in particular, I wanted to comment a bit more on the dynamics of the structure function coupling that you investigate. So the temporal dynamics aspect, which I think is very interesting. So you report that basically unimodal regions show stable coupling over time, while transmodal regions fluctuate more. Exactly what you're showing now in this slide. And I mean, these basically reflect a lot the values of SDI as well, right? So the regions that with lower structure function coupling appear to be the one that are more stable. So I wanted to ask you, here you are computing standard deviation on the SDI or on the log. I mean, I assume on the SDI from what I read on your manuscript. So how do you think that basically your measure of variability might be influenced by the fact that the range for coupling is smaller? I mean, coupling indeed as a definition is between 0 and 1, so it has much smaller values, right, than decoupling that is higher than 1. So you see it as a problem, this asymmetry, or?
Thank you for the question. So to give you like a little bit more detail there. So basically we adapted the STI characterization to take the time aspect into the account here, in a sense that we computed the STI within 1 second window over time. Okay, so that's like the first step towards the time resolved analysis. So once you compute the STI for each of the 1 second chunks, you get the time resolved STI values. And then on top of that, you compare, you quantify consider the standard deviation as the index to quantify how much the STI values vary over time. So that's in the end, you get like a standard deviation map across the entire brain, and then you threshold, take top and bottom 10 percentile to see which regions exhibit more temporal fluctuation and the minimal temporal fluctuation.
Yeah, this is clear, exactly. And this is the result of your, I mean, what you're showing here is the standard deviation. But my question was, I mean, how do you interpret the regions that you obtain? You see, for example, the ones that have low temporal fluctuations are the ones that are more coupled, right? So do you think this is a true interpretable results, which would mean, they're more unimodal, they vary less probably because they are all the time more coupled to the structure? Or how much do you think it is influenced by the fact that the range of coupling is between 0 and 1? So obviously these are smaller values, which might be related to smaller standard deviations as a consequence, other than decoupling values that can be much higher. So how much of this could be related to an artifact due to how you construct the SDI measure? Or not, maybe. I mean, what is your thought on that, and how could you solve it in case?
Okay, thanks for the question. So the interpretation of this is that one potential way would be is that these if you look at the regions that exhibit minimal fluctuations, they constitute unimodal systems. It could be because of the functional specialization there, in a sense that they are inherently functionally specialized to one modality as compared to the rest of the ones. So it is not so egregious that it exhibits minimal coupling as compared to more transmodal ones that are multimodal, but not functionally specialized in nature. So as far as the values are concerned, so 0 to 1 and basically sub-zero and the over zero, they represent decoupling and coupling, right? So when you quantify the coupling there, okay, I see why you say that, in a sense that there is more coupling than the decoupling here. Yeah, it could be that there could be something. So
I mean,
even though, sorry, if you want to add something.
no, go ahead.
Okay, so here what we are looking at is essentially the relative fluctuations, right? So I'm thinking that it would not impact so much, in a sense that you have a distribution between 0, some -2 to +2 or something like that, and then we clip it to -1 and 1. I believe I did not clip for performing this time resolved analysis. And end of the day, we are looking at the relative fluctuation in the regions. So in that sense, I believe this holds true since we are going with relative fluctuations. I'm sorry if it, I'm not sure if that answers the question, but yeah.
I agree that this could be a realistic result, as you say, like, I mean, it could be totally believable, right? That these regions are more constant because they kind of have always the same role and being more attached to the structural connectome. What could you do in order to rule out this asymmetry and 100% be sure that, I mean, as an additional step, I would do it just as a safety check, right?
Yeah.
What could you do? Because I think over your manuscript, you do it already for some visualizations when you visualize the SDI. What do you do when you visualize it in your figure? In order to have a symmetric range,
Yeah, symmetric range is basically that is enabled by clipping the specific endpoints, right? Like -1 to 1, for instance.
Yeah, but in order to have -1, I think you do some operation, right? Because the SDI wouldn't be negative otherwise.
Yeah. So basically, when we once we apply binary log, they extend up to 0 to 1, right? Because it's just before computing the ratio. After computing the ratio, if it is 1 higher than 1, that represents that's like basically the 0. Yeah, midpoint, right? So right when we apply binary log, it goes between negative to plus ends, right? And then on top of that, we clip to be able to make it symmetric. So that's one of the ways we approached for the study 1, because it's not a contrast condition. And for study 3, we did not perform clipping because of the contrast conditions there. So yeah, and it's a really good point, and I have to really think about like how to rule out that it is some of the effects that we observe is driven by the the disproportionate coupling versus not so much in the decoupling part.
Yeah, as for the visualization, I think, as you say, I mean, when you apply the binary log, at least then you have a similar range for coupling and decoupling. And then, I mean, just as a safety check, I would maybe try to apply it and see what you get. Even if, as I said, I mean, this would be totally realistic result. Then maybe another question always related to this study, if you go to the slide before, I think.
Yeah.
Yeah, so when you look at your spatial maps of basically SDI in movie watching and resting state, so you get very, very few regions that are decoupled, right?
Yeah.
I mean, you can see it from here, but in any of the conditions that you look.
Yeah.
How do you explain that? Is that like a consequence of your thresholding because you also apply the null models, and so these are, I guess, the significant SDI values that survive?
Yeah.
Was there something more in the original map that is not thresholded, or why is it that so few regions and so focused appear as decoupled in your?
Yeah. Thank you for the question. So first step, once we compute the graph Fourier transform, we get the graph for spectral density, right? So there we have like essentially steepened 1/f in a sense that increased like pronounced low frequency graph power compared to the other aspects. So we have tried like a different manner to bifurcate, dichotomize the spectrum to be able to determine the the coupled harmonics versus the decoupled harmonics. And despite even choosing, for instance, 2 or 3 the cutoff, like median cutoff to choose connectome like coupled harmonics and the decoupled harmonics, we observed a similar topography. It goes to show that the activity is like more coupled, strongly coupled to the structure, meaning brane-wide coupling. This could partly be explained by the nature of EEG signal and the source localization smoothing parameters there. However, because when we compare against the literature which have used different source localization inverse modeling with different smoothing parameters, it doesn't necessarily rule out on the smoothing front. It appears that there is the volume conduction plays a massive role with the EEG side. And if you look at the MIG, for instance, so if you look at the median frequency, critical frequency of MIG, for instance, which is not so much affected by volume conduction, there is a pronounced there is a clear shift in the critical frequency, meaning that it's not low frequency dominant. If that answers your question.
Okay, now we see.
Sorry, wait, I'm going there. Yeah, this is the one. So yeah, if you look at the just to add to my last point. So yeah, if you look at the distribution of the critical frequency, median frequency here, there is a clear shift. It goes up to like 20 to 25, eigen modes, suggesting that there is a dissipation of graph spectral power by nature in MEG as compared to low frequency dominant EEG signal there.
Okay, maybe a last question for now. If just again on the meta-analysis that you did with Neurosynth for the first study, maybe can you take us once again through the interpretation, because it was not 100% clear to me here.
Okay.
What you want to show as a difference between the two conditions or as a finding, because it's true, I mean, you want to compare it with Margulies gradients, But because there was like a diagonal, right? So
Yeah.
shows like a macro scale going from lower level sensory to higher level cognitive regions. But here it's totally different, right? So what is your take-home message from this analysis, actually?
Yeah. So indeed, we do not have like a nice gradient as in Margulies' work, and even in your 2019 STI fMRI work, there is a nice gradient. Here, it's more specific in a sense that the unimodal regions. So first of all, we are trying to map the spatial map onto large scale functional domains to extract the distribution of the intensity of coupling to decoupling. So that's the key idea here, and we compare between two conditions separately, meaning that we perform meta-analysis for video watching first, and then resting state another, not a contrast, not like contrast and compare and perform meta-analysis. So the interpretation is that there is a weak gradient, so to speak, in a sense that unimodal strongly anchored on the coupled side still holds, but there is a specificity in the higher order codices. Instead of like anchored on the coupled end strictly, it spans between coupled to decoupled axis. So the interpretation here is that there is a transmodal codices exhibit a weaker coupling, not necessarily strong decoupling, but rather a weaker coupling as compared to unimodal codices exhibit strong coupling. And this pattern is consistent between movie watching and resting state.
Okay, so here you wanted to highlight basically similarities between the two, because they are quite similar apart from some differences, right? Okay. Okay, thank you.
Thank
you. So now we're gonna move to Lune Bellec, representant of the Dean.
Yeah. So as the Dean represented, my role here is to ensure that the protocol is followed. I cannot say anything of the French part. It's very complicated gathering today. I will comment only on the University of Montreal side. Everything seems conform. I just noticed a lot of loud applause at the end of the presentation, which is a bit louder than we would normally tolerate at the University of Montreal, but I think it's okay. Maybe reflective of the quality of the work. So we shall proceed. That's all.
Yeah. It's not a question?
No. Oh, no. No, I'm here to serve.
You can ask question if you want. So if you don't have question, now it's technically my turn. For the sake of time, I have just two little follow-up questions because I had like actually Celine and Enzo who asked my questions initially. So the first one was related to the variation of the DWI and use the personal DWI versus template. You already a bit addressed that, but I was curious about where in the harmonics and anatomically there's the most heterogeneity in terms of the variations of the WI and the harmonics.
If I were to venture into guessing, it would be more pronounced in the heteromodal, transmodal cortices, I would think.
Okay.
Yeah. So I haven't really followed the literature that would back me up. But if I were to venture into a guess, so that would be that.
And do you think it correlates with the heterogeneity of phenomenological experience?
Heterogeneity of, sorry?
Of the phenomenological experience?
Mm. So you're suggesting that whether the changes in terms of structural connectivity in the heterogeneous transmodal codices would correlate with the phenomenological aspect. Is that right? Okay. So studies have looked into structural changes like diffusion changes under LSD. If you look at the time scale, I don't think we are seeing massive differences while subjects are in the LSD in the diffusion ones.
No, I was not meaning to that. I was more saying like, would you guess that the variability of phenomenological experience of people can be correlated to the variation of neuroanatomical structure? Let's say you have more connections between your auditory cortex and your visual cortex, and you will be more prone to have like synesthesia under LSD, for instance.
So I haven't come across any particular study that would explain this. However, if I were to guesstimate, it would be that changes in the neural, so neuroanatomically speaking, then it depends on which kind of metric that we use to define the structural connectivity matrices, for instance. Here we used density, for instance, meaning that number of connections divided by the taking the volume into the account. And it depends on which the metric that we are using, but I wouldn't necessarily think that this could explain the various phenomenological aspect precisely.
Okay. And the other question was more associated with the correlation with receptors. So typically you show that the motor regions were less impacted. Did you try to explain how much the impact of LSD was correlated to the density of 5-HT receptors?
Yeah. So wait, where is that? So let me put that. Let's go. Yeah. So this is the gradient of the effect here, right? It does look like tracking the receptor density in a sense that wherever we are having like maximal disproportionate expression of 5-HT2A receptors, there are maximal drug effect. We performed the analysis after it was super interesting, and I went and looked, like compared between the two, but there wasn't a significant regression, significant correlation between the two, like a minuscule correlation, and that is like potentially, again, the similar story, same answer, limited by the sample size. It wasn't very convincing to move forward with the interpretation.
Okay. Thank you. Yeah, that's gonna be all for me. I don't want to take too much time, so we can give also the opportunity to ask questions to the supervisors. So maybe started with Julia Lioi.
Yes, thanks, Guillaume. Well, I don't think I will ask question to Venkatesh, but if I may spend a few words for him to congratulate him for all this fantastic journey, especially because I didn't have the opportunity to be there with him today to support him in person. So I just want to say to you, Venkatesh, I'm really proud of you and how really did you progress during these years, how you were able especially to face all the ups and down of a thesis far from your family and sometimes from some of your supervisors and your friends. I'm really amazed at your enthusiasm, your strength, and your kindness from the start to the end of this PhD. Well done to you, Venkatesh.
Thank you, Hina. Nico?
Right. So, Venkatesh, you know that I have a question, and it's the same question that I keep asking you from time to time. That's been a kind of almost a joke, but also like something that we like to play with. But I'm not really going to ask you this question because this question, as you know, is so Venkatesh, how do you think the brain works in the end? So, I mean, I don't expect you to answer this question, neither that I expect anyone to really be able to answer this question. And from what I've seen in your final presentation today, with like the different evolutions of this presentation and the different evolutions of the way you were presenting all across the years, I can say that you start to have a bit of some starting points, interesting things to say about how the brain works in the end. So congratulations about that. Your journey is a very particular one. I just will recap a few things about what we know about you, because we got to know you 5 years ago. You were a master's student in data science in our school, which from the starting point doesn't really how can I say predict anything about becoming a cognitive neuroscientist. So the challenge that when we started to know you was that we had an intuition that you wanted to move towards becoming more a cognitive neuroscientist than a data scientist. So I don't know if the challenge has been fully, if you succeeded in that challenge. That's not me to say that, but the committee and the jury. But it's I'm personally, as Julia, very proud of what you've been able to achieve along those years with the different facets of the different ways that you saw, that you investigated the questions. And of course, there's more things to do. I'm sure you will continue in this path. And I hope that despite the different things that happen and the ups and downs and things that sometimes in every PhD does things like this that happened, I think you manage very well. So congrats for that, and looking forward to discussing more and seeing what's for the future. Thank you, Venkatesh. Thanks.
Yeah,
So well done on your presentation today. I think it was really, really good. And I just gotta just for a quick few words. I wake up either very early in the morning or I go to bed very late. This morning I woke up really early, and what I did, I went to our Slack exchanges over the last year. I downloaded all of them and I gave them to an LLM. And I explored some of the things that LLM was telling me about your journey. So I'm just gonna very briefly tell you the summary that comes, and this is obviously biased by whatever we say on Slack and we don't say in real life. But basically, Venkatesh's PhD has everything a proper Coco Lab adventure needs: administrative side quests and issues and problems with the admin, funding cliffhangers and issues with delayed funding, manuscript marathons, reviewer battles, journal strategy debates, and many deep dives into what LSD does to brain dynamics, deep dives in literature, but also in real life. Along the way, Venkatesh mastered both electrophysiology at the fine art and the fine art of surviving university bureaucracy. He explored peak frequency shifts, aperiodic slopes, complexity, connectivity, and all the interpretive dangers hidden between interesting finding to do not overclaim. There were figures to polish, abstracts to rewrite, journals to target, postdoc plans to imagine, and finally, in true PhD fashion, one last crucial methodological question in exchanges with Karim: which room has the best setup for the defense? So from Slack chaos to scientific clarity, from funding delays to today's defense, Venkatesh's journey has been rigorous, collaborative, occasionally psychedelic, and very cocolabby. So now, this is now from me. I'd like to congratulate you, Venkatesh. I'm also proud on your scientific rigor, the hard work, the determination, the curiosity, and the audacity to follow your interest in altered states of consciousness, and also for navigating with impressive patience and grace the tricky complexity of having 3 co-supervisors over different time zones. So that's been an additional challenge that adds on top of the administrative challenges of having this co-tutelle thèse, changing departments and all of that.
Yeah.
So that adds on top of, so this also gives more value to all of the scientific rigor and research work that you've been able to achieve. So congratulations on that.
Great. So if there is no other round of questions from the rapporteur and external members, we can switch to the committee discussions. So for those who are on Zoom, I sent you the link for the other room associated with for the. So maybe we can let that run, but cut the sound,
And cut the
and
stream.
we're gonna move to the
Okay.
on my laptop on the other Zoom.
Yeah, okay, that works.
Yeah,
So you
exactly.
could connect here. I have to stop this.
You can just cut the sound,
The sound should be fine, right?
and I'm gonna ask the people to.
So do we leave this Zoom room and connect to the other one?
Which one? So did you send my email or?
I
you
think
have
he
to-
sent 2 links in the morning, so
Uh,
it's
also
I
by
see.
email.
Uh, I sent this morning. There is like.
Oh, wow. Okay. Yes.
Yeah, I guess Julia, you can leave as well.
C'est bon.
Bon. Julia, si tu
Yeah.
m'entends, il faut
Yeah,
que tu
good.
passes sur l'autre.
Just waiting for Julia.
Je vais montrer les gens, Guillaume.
Ouais, vas-y, allez.
Yeah.
So,
On YouTube.
qui veut le...
C'est le président de jury.
Ah, c'est moi qui dois...
Non, c'est la liste.
Donc, c'est à moi qui revient le...
On va lire Bon, normalement, on lit le rapport, mais bon, on lit les éléments de rapport. Enfin, comme tu veux. Non.
Et on va devoir avoir le serment de choses.
Ah, il y a un serment.
Oh, le serment.
Ah, oublié le serment, d'accord, mais c'est pas possible. À chaque fois, tu oublies ton serment, toi.
Ah non, au mail, on l'avait pas oublié.
On l'avait oublié, on avait dû le chercher et ça s'était fait en panne.
On avait le serment.
On avait le serment cette fois-ci.
The oath. The oath also. You have it. I don't have it. Ah, you need to.
Do you know about it?
You know about it. Yeah, but okay, so you need to get it. Yes, yes, to get it. We need to get the oath. So,
But before that,
we need to get the oath, but you need to have the
one of the
text somewhere.
pleasure to be a president is...
Okay, wait.
On en
Dr. Subramani.
Exactly. And for the oath, I leave you the... You have the text?
I will find it quickly. Okay.
Juste prendre une petite photo.
Yeah, we need
On
to
peut faire une photo.
de serment.
Maybe we can put
Yes,
the...
maybe put the
Everybody...
Ouais, c'est ça. Comme ça, il y aura
As,
tout. Ça va faire un petit souvenir. T'arrives?
like a gallery like this.
C'est la deuxième fois que je fais une thèse co-supervisée France-Canada. C'est la deuxième fois qu'on perd le serment.
Je l'ai peut-être encore, le serment.
Tu l'as encore, Maël? Aïe, aïe, aïe. Non, je vais le trouver sinon.
De mémoire.
Ah non. Oh, there you go. I have it. Wait for it.
Venez pour la photo déjà.
Serment des docteurs. There you go. Oh, it's here. Do you want to read it in French or in English? Both works. Anything else?
Maybe in English.
Maybe
Are
in
you gonna
English.
show this to your family?
Because you will have to tick a box as.
Yeah.
Ah, you need to.
So, and the interdisciplinarities.
Okay, interdisciplinary.
Yes.
Okay,
No, interdisciplinary.
in
Interdisciplinary.
okay.
Yes.
So starting with in presence. Okay. So in presence of my peers, with the completion of my doctorate in interdisciplinarity, in my quest for knowledge, I've carried out demanding research, demonstrated intellectual rigor, ethical reflection, and respect for the principles of research integrity. As I pursue my professional career, whatever my chosen field, I pledge to the greatest of my ability to continue to maintain integrity in my relationship to knowledge, to my methods, and to my results. And that's it. Yes.
Will somebody take a picture of the member of the juries with the background?
The people in the background.
People in the...
Well, everybody.
Photo.
So, yeah, with members in the background? Ça les dit peut-être que deux par là.
Ouais,
par là, ça va. Peut-être, je sais pas si tu l'as pas bien.
Yes.
Yeah.
Okay. Congrats. Yay.
Okay. The first, yeah. Yes.
Thanks for hearing them. Fortunately, the rest of the members of the jury are not here for the rest of the fun. But we will think of you.
Yeah, thank you very much. I will contact you for the rest of the paperwork, but thank you so much for your flexibility and efficiency and your presence even virtually.
Thank you.
Take care.
Thank
Congrats again. Thank you.
Congratulations.
Bye-bye.
Congratulations.
Thank you.
Bye.
Merci, thank you.
Merci à tous. This is a gift from
Merci. Thank you so much.
Ciao Venkatesh, bravo. Congrats. Bye guys.
Julia says, Julia, bye-bye. We'll catch up.
We'll talk
Bye.
later.
Yes.
Well
yes.
done to you. Have fun and rest.
Ciao. Bye.
Ciao. Have fun.
What? Thanks a lot. Wow. Oh wow.
On est allé vite, mais il y avait quand même 3 super.
Should I stop this? Okay, I'll put it congratulate this guy. Bon, mais du coup, je vais faire ça ensemble.